Hello everybody, this is my group's project about the effects of sound and light on the reproduction of Drosophila melanogaster, otherwise known as fruit flies. In addition to the experimental data being presented later on, we have provided some background information on the factors that we are going to talk about, namely light and sound. We have researched that light cohabitates with the heat of these flies' ideal environment in nature, as it is stated in the slide that 25 degrees Celsius or 77 degrees Fahrenheit is sort of where they would reproduce at a higher rate than the other temperatures. But we have also figured out that if they're placed in an area where the temperature is higher than 25 degrees Celsius or 77 degrees Fahrenheit, they would experience heat stress and develop much faster. However, in exchange to developing much faster, they have a more weaker body compared to those that don't experience heat stress in these hotter temperatures. In addition to light, we have sound. Now in this case, we also realized that the humans and fruit flies have very similar hearing. When placed next to a loud sound producing device, they also experience the same discomfort as we do, and also has the effects of impaired hearing similar to that of a human. Now, the purpose of this experiment was to see if different kinds of light and sound exposures could help improve invertebrate reproduction in endangered species such as the bees, which are dwindling every year. The two hypotheses we have came up with for this experiment is if there's a higher light intensity or exposure to the flightless fruit fly colony, then there would be an improved rate of their reproduction, and the same thing would happen to a higher sound exposure. Now there's the four variables that we have described below. The independent variable would be the lamp or speaker placement, as we've chosen 4 factors, one at 0.6096 meters or 1.2192 meters. Then for sound we have 30 decibels and 50 decibels. Now the dependent variable would be one of how much of these fruit flies were created or how many eggs they produced. The controls would be that there was a fruit fly culture that we placed with no extra light or sound conditions that we have set upon them. And the constants would be the little habitats that we have set up for them in the soup containers, and the amount of food that are given, and the amount of the sort of excelsior that they use for their living space. Just a quick overview, here are the materials that we used for the experiment. We had some speakers that we used to mimic the sound. We have some lamps that we used to mimic the light source. And as you can see in figure 1, the container setup, there is a green concoction on the bottom that is a fly medium, and on top that is the yeast to prevent mold infection, so it would not hinder our experimentation. And in figure 2, which is the container top, it has these blue sort of color on the holes. We have figured out that if we put the blue colored Sharpie on top of it, they tend to stay away from these holes, which makes it a lot easier for us to handle them during this experiment. In regards to procedures, we had 1 control group, 2 experimental light groups, and 2 experimental sound groups, and we recorded the data every 4 days regarding the amount of male fruit flies, female fruit flies, total amount of fruit flies, and eggs produced between a 95 centimeter squared area. And then we would get rid of the old containers and make new ones to be put in their place after we recorded the data.
In graph 1, change in the amount of flies after the initial 4 days, you can clearly see that there is a difference between the 4 categories that we changed from the control compared to the control. In it, you can see that the change in light 2 feet is much higher than the control, which supports our hypothesis that the light will positively impact the control, or the amount of flies in reproduction. But you can also see that the change in light 4 feet has actually a less increase compared to the control, which goes against our hypothesis. However, you can also see that the change in sounds, both of them have a lower average than the control, which does support our other hypothesis that the sound will negatively impact fruit fly reproduction. Table 1, the change in the amount of flies after the initial 4 days, just shows these values without the standard error in table format. Graph 2, control group day 0 through 36. In this graph, you can clearly see the data collected from day 0 to 36 for our control group. The data was collected every 4 days, and in it you can also see our standard error. In this data, you can clearly see that after the 0th day, every 4 days, the amount of flies counted averaged from between around 59 to 65. Graph 3, change in light day 0 to 36. You can clearly see both of the data collected for both the light fixture being 2 feet above the container, which was shown in the blue, and the light fixture being 4 feet above the container, which is shown in orange. In it, you can clearly see that there's a very high average for the change in light 2 feet above the container, with it averaging between 90 to 95, whereas the change in light 4 feet above the container, it averages from between 55 to 60, which is a very high difference between the two values. In graph 4, change in sound day 0 to 36, you can clearly see both our data points for the change in sound 30 decibel music, shown in blue, and the change in sound 50 decibel music, shown in orange. In it, you can see that the average for 30 decibels was between 50 and 60, and the average for 50 decibel music was between 45 and 50. In graph 5, all categories, total amount of flies from day 0 to 36, you can see all 5 of our categories, with the control being in blue, the light fixture being 2 feet above, shown in orange, the light fixture 4 feet above, shown in gray, the speaker playing 30 decibel sounds, shown in yellow, and the speaker playing 50 decibel sounds, shown in light blue. In it, you can very clearly see that compared to the control, the 30 decibel music, 50 decibel music, and the light fixture being 30 feet or 4 feet above the container was actually lower than the control. Not by too much, but the 50 decibel music had at least a 20 fly decrease compared to the control, whereas the light fixture being 4 feet above or 2 feet above the container, you can clearly see that there is a much higher amount compared to the control, where we did nothing, of from at least 30 to 25 flies increase, which supports our hypothesis that light would impact it positively.
In conclusion, our first hypothesis was completely supported. However, our second hypothesis wasn't. As we can see, there was a effective way to hasten the reproduction rate when it came to light sources. For those that we experimented on, they preferred a closer one, as their population increased to about 150% than that of the control group, and so did the egg production rate, which was 279%. However, when it came to loud sounds, like around those of 50 decibels, it was quite ineffective, as they kept on decreasing every time that we would check up and analyze the data, and it would decrease by 25% compared to the control, and around 22.7% for the egg production rate. There were many limitations when it came to this experimentation, as we did it outside, and due to a fear of having a fruit fly infestation if we did it inside. In doing so, there could have been many external variables such as the wind or even the temperature that could have caused more varying results. However, this experimentation can also have a lot of applications, as it provides more data to the already limited information known about fruit fly hearing. Thank you for your time on listening about our project regarding the effects of sound and light on Drosophila melanogaster, otherwise known as fruit flies.